Hi everyone. I see there are people still joining, but we will start. Thank you so much for joining us today for our Sterilization Monitoring Essentials webinar. My name is Calibri Ntlokwana, and I'm the Product Manager for the Food Safety and Microbiology Division at LASEC. And joining us today is Rick Daschner, who is the Director of Sales and Marketing at True Indicating, and he will be giving us our Sterilization Monitoring Essentials presentation today. So we've set up this webinar today because one, sterility compliance is of great importance to a plethora of industries, but most importantly, we've noticed a knowledge gap in terms of the tools and procedures that are needed in order to maintain those A-grade sterility standards and on a more routine basis. So this webinar answers exactly that. What are the essential tools and procedures that you need in order to improve your sterilization monitoring procedures and thus in turn improving your sterility compliance to a gold standard? We are all set to begin, and I'll be handing over to Rick shortly. But just a quick note, you will see that your mic is muted and the chat is disabled for this session, but we'd love to hear your questions, so please do go ahead and submit them in our Q&A section. And after Rick has done presenting, we will try to get to as many as we can. Over to you, Rick.
Oh, thank you, and good afternoon. Yeah, as was said, I am Rick Daschner. I'm the Director of Sales at True Indicating. We are located in Toledo, Ohio, USA, and we manufacture all of our both chemical indicators and biological indicators at our facility in Toledo under one roof. Just quickly, as it relates to chemical indicators, and you'll learn more as we go on. These are a product almost kind of like a label or a sticker in a lot of cases with an indicating ink on those. And we manufacture all the inks in-house and we print all of these in-house. So we control the entire process, which is a quality standpoint and a delivery and availability standpoint. So I'm going to start with the presentation. My goal is to first talk slowly. I've heard I talk too much too quickly. So that's number one. But number two, of course, is to pass along some education as to how our products can be used in your environment. And if you're not sure on some things, again, set up the questions. Like you said, we will get to as many of the questions as we possibly can by the end. But if we don't get to all of them, I promise you, we will get you those answers after the presentation at a later date, probably via email or in some other fashion. So without further ado, let's get started. So understanding sterilization monitoring is the first section here. And just to make it clear, sterilization, this is the process of eliminating all forms of microbial life, including bacteria, viruses, fungi, and spores from the surface, liquid, or biological culture medium. This ensures that an object or substance is completely free from any living microorganisms. Again, I want to stress that sterilization is basically designed to kill everything, where disinfecting is maybe not going to kill things at that level. And sometimes sterilization and disinfection get confused. So be focused on the fact we are talking about sterilization here. So sterilization monitoring. So we have a process of killing everything, but how do we know that it actually was effective? So sterilization monitoring, this involves the checking and verifying that sterilization process has been effective. Monitoring can include using biological indicators, such as spore tests, chemical indicators like color changing strips. And again, these are the products that we're going to be discussing, and these are the products available to you to verify that your sterilization processes are effective. So what's the importance? Again, it's going to depend on the environment that you're using these in, but preventing infections in a hospital or medical environment, and also potentially in a manufacturing environment. But sterilization helps in preventing the spread of infections by ensuring that medical instruments, surgical tools, and hospital environments are free from harmful microorganisms. Now, in addition to that, that would also be implants. So anything that's going to be put into a patient needs to be sterilized. Patient safety, it ensures patient safety during medical procedures, reducing the risk of complications and infections. compliance and standards, and that was brought up earlier in this. There's a lot of standards and compliance issues that need to be addressed and taken care of. And the one thing that's nice about the indicators is this will achieve that goal. Healthcare facilities must comply with stringent health and safety regulations that mandate effective sterilization practices. And then again, protection for healthcare workers and potentially protection for other workers in a manufacturing environment. It minimizes the risk of healthcare workers contracting infectious disease from contaminated instruments and surfaces. So what is the importance of sterilization in other industries? And I alluded to that earlier. We do quite a bit of business with pharmaceutical companies, and we offer a range of products that are used in actually a range of different areas within the manufacturing and sterilization product process of their products. So pharmaceuticals ensures the sterility of drugs and medical devices, which is vital for the safety and efficacy of these products. And that's one of the bigger ones. And again, we are pretty prominent within many of the major pharmaceutical companies all over the world. Food and beverage. Sterilization process in food production prevent foodborne illness by eliminating harmful bacteria and pathogens. Again, it's something that we want to, the manufacturers want to make sure when these products get out to their customers that they're safe. Cosmetics prevents contaminations of cosmetic products, ensuring that they are safe for consumer use, and a wide range of uses in the biotechnology and research areas to maintain the integrity of experimental results by preventing contamination in laboratories and research facilities. Now, when we're talking about biotechnology and research facilities, their uses and ranges and their creative ways to use our products are all over the place. So to answer like a question of how are they used, it's all going to come down to whatever they're manufacturing, whatever their project is, and what their goal is as it relates to sterility. So we make a host of products, which you'll see when we go forward here, that will fit those needs. So methods of sterilization. Now, sterilization comes in several different methods, but one of the probably the most popular and most prominent would be steam sterilization. And steam sterilization, commonly known as autoclaving, is widely used method of sterilization that employs saturated steam under pressure to achieve high temperatures. This method is particularly effective because the combination of steam and pressure can penetrate and denature proteins, effectively killing all microorganisms, including spores. So, and that's what you're going to see in most doctors' offices and hospital settings. Advantages, it kills a broad spectrum of pathogens, including resistant spores. It's versatile, can be used for a variety of instruments and materials, except those sensitive to heat, high heat and moisture. And we'll talk about what you do with those products going forward here. But so the key elements of this are primary agent is heat and moisture. Pressure is used to increase the boiling point of water. And temperature standards include like 121 C, but sometimes up to like 132, 135, 137 C, depending on the product. And our products will be validated to a couple different temperatures that will accommodate what you're doing, what you need. Dry heat, just like it sounds, dry heat typically requires high temperatures and much longer exposure times compared to moist heat, like autoclaving. And just to give you an idea, again, the differences, and you're talking minutes versus hours when you talk about this, comparing it to steam. But standard temperatures 160 C, 170 C, and 180 C. Now, dry heat works by oxidizing cell components, leading to the destruction of microorganisms. And you can kind of see the pictures off to the side. Kind of looks like an oven in some cases. That's a pretty rudimentary dry heat unit. And then other dry heat units, you're not going to see the elements. And that's like the more popular ones below for instrumentation. Ethylene oxide sterilization. Ethylene oxide has come under fire lately for its damage to the environment and concerns of communities that have ethylene oxide processing plants and usage nearby. So it's a great way to sterilize, but it has come under fire a little bit. So keep that in mind. But it's still used widely. So EO sterilization is a method that uses ethylene oxide gas to sterilize medical devices, surgical instruments, and other items that can't withstand high heat and moisture. It's especially valuable for sterilizing complex and delicate devices, as well as heat sensitive materials. So when you have a project that's a plastic or maybe even electronic or something that's going to be damaged by moisture and high heat, ethylene oxide is an alternative. So the way it works, it has an active agent that disrupts the DNA and proteins of microorganisms, effectively killing them. And you can see the temperature is much lower, 37 C to 63 C. So a lot lower. And exposure, again, though, you're up in the hours with something like this. So you're not going to turn something around quickly in a project or in a hospital environment with something like this, where the autoclave is much faster. Hydrogen peroxide gaining a lot of traction now. Companies out there are producing hydrogen peroxide sterilizers that are effective and becoming more affordable. So VHP sterilization is low temperature sterilization method that uses hydrogen peroxide vapor to sterilize medical devices and other sensitive items. So the key elements, again, hydrogen peroxide vapor, the active agent that disrupts the DNA and proteins of microorganisms, effectively killing them. And the temperature is low. So you can see 28 to 40 C. And deep vacuum process involves creating a vacuum. Oops, I'm sorry. Whoops, did I miss? Creating a vacuum to make the have it better penetrate. I need to go back. Sorry about that. There we go. To ensure that penetrates the surfaces. Now you'll see on the top right over there, that's a storage scope. Scopes are within the hospital setting are very delicate and very expensive. So if you can use some sterilization method that won't damage these, because they are reused, and the scope obviously is used within the human body, they need to be sterilized and sterilized properly without damage. So that's why you see a little picture of the scope up there to show you that. Radiation. Now, radiation's interesting because it's becoming very popular. They're popping up all over the place for all different types of industries, and there's a couple different approaches that they can take for a radiation sterilization. But radiation sterilization is a process that uses ionizing radiation to eliminate microorganisms from medical devices, pharmaceuticals, and other materials. That this method is effective for items that cannot tolerate heat or moisture. Now you're saying, so there's other ones that we just talked about of the same vein that can't tolerate heat or moisture, but why radiation? And one reason for radiation is it can radiate large loads of product, pallets of pharmaceuticals, pallets of things. And that's one of the reasons it's becoming popular from a commercial standpoint, is that it can handle a lot of product. Now, there's different types of radiation that are used, and all have their positives and negatives. I'm not really going to go into them onto this presentation, but some of them are used for different reasons. But gamma rays, which are high energy photons that penetrate deeply and effective for large bulk materials. Again, like I said, they can run pallets through. If you see the picture on the right there, that's a commercial facility that runs these giant pallets through. Electron beams, or E-beam, as you probably hear, high energy electrons that are less penetrating than gamma rays, but effective for surface sterilization and thin materials. And then X-ray. X-ray is becoming more popular. There's not a lot of indicating products out there that are validated for this. I'll be, right now, our radiation material is not validated for X-ray. However, it does work and it is used for validation by companies that have done their own internal testing of it. But we have not yet, because it hasn't really gotten the traction yet for us to put the resources into validating for X-ray. But our radiation indicators will work. And what we'll do in a lot of cases, or if you've got these facilities available to you, or you have a third party that you might want to talk to about sterilizing your product, we would be more than happy to send them a few samples they can run them through and to get the confidence that they need that these would work. But similar to gamma rays, but they're produced differently, they provide deep penetration for bulky items. And that's the draw for that. So what does True Indicating have to offer? Let's start with chemical indicators, and we'll kind of walk through the differences in chemical indicators, the different configurations. And also, if you don't see something that you think you might like, we are a very flexible company who, for the right size volume, can customize just about anything that you might need. So let's walk through this. So chemical indicators. First thing is, and I saw some of the questions that had come in earlier, kind of asking, what the benefits or how they might work together, chemical and biological and things like that. Let's kind of walk through what chemical indicators are used for. And the biggest one is process indicators. And again, it's just to say, yes, this was run through a sterilizer, or no, it wasn't. And that's what these are for. It's not going to guarantee that everything in there is sterile, everything worked. It just says, yes, we ran it through the sterilizer. And typically these are placed on the outside of packages. They change color when exposed to the sterilization process, indicating that the items had been processed. Again, that's what they do. Internal indicators. Now, these are going to be placed within containers to ensure that the sterilization process did make it into, let's say, the inside of a package. And they're placed inside packages or containers. They verify the sterilization, steam, ethylene oxide, hydrogen peroxide has penetrated or reached the items inside. Again, it's one of those things where it gives additional confidence that the sterilization process had reached the product that you're trying to sterilize. And we'll go into why in some of these questions, that there were some good questions that we have some answers for you, how that might work to give the confidence that the sterilization process was validated. Now, integrators are a chemical indicator. They look just like all the other indicators in terms of type 4s and type 5s and 6s and things. We'll talk about that. But they provide more detailed information by mimicking the conditions necessary for sterilization, changing color when all parameters are met. And that is time, temperature, and exposure are met. And then emulators, or our type 6, that is almost a more stringent than a type 5, which was the integrator. And that's similar to integrators, but the design is designed to simulate specific cycles of sterilization process that offer precise validation, specific types of, and with specific types of loads. And that one is, again, it's one that used in conjunction with a biological indicator can be really effective. So what does True Indicating offer? There in front of you is the list of all, let's say, the process type 1 indicators that we offer. Now, type 1 indicators, as I had mentioned, are indicators that say, yes, it was processed, or no, it wasn't. And you'll see we've got steam dots, which are usually just stuck on the outside of something, and or can be put inside. Ethylene oxide, the plasma or hydrogen peroxide, the radiation indicators, and dry heat. Now, you'll notice on the dry heat, just to give you an example, and I talked about potential customization, and we made this particular label on the bottom right for a company that wanted a little more detail on it. They wanted more of a label. They wanted to write the date on there and other information. So we created these for them and found that this was popular with, quite a few other customers. And so we made it a standard product. But these are the kind of things that keep in mind to think of if you have a need for this, we can make that customization of any of these products to include areas to put down data. So tapes. Obviously, tapes are popular because they serve two purposes. One, you can tape up blue wrap or other packaging material to close it. But two, it will indicate whether or not it was processed. And that's what's really nice about the tapes, because you kind of get a 2-in-1. So steam tapes, autoclave tape, as it's most widely known, we sell a lot of, very popular. Next would be ethylene oxide tape. And then what's becoming more and more popular is our hydrogen peroxide tape. And just one thing to note about ours versus others, this is a plastic tape, because hydrogen peroxide, you have to put on more of a different type of substrate, and more of a let's say more of a plastic, more resistant tape. This tape, what we call easy tear tape, is serrated. So I don't know if you've ever worked with tape before, that this type of tape, but you've got to find a pair of scissors to cut it, because you cannot tear it. It will not tear. However, since ours is serrated, you can tear off what is needed, tape up whatever you're taping up, and then just tear it with your fingers. And it may not sound important, but to the people that have to deal with this stuff and trying to hold the tape down, find a pair of scissors, or do whatever, it is a huge advantage. So, and we are the only ones that make this, and it's a great time saver for you. So type 4s. So we've gone from type 1s, and type 1s, again, was it processed? Was it not processed? Type 4 adds a couple more parameters to it, and it's designed to react to 2 or more critical processes, time, and then temperature and moisture or moisture as stated values. So our stated values on the product, you can see what it is on our data sheet. And this will give you a little bit more of a response to say, yes, it was processed, and yes, it did meet the temperature or moisture parameter. And these are, again, type 4's indicators are frequently used for pack release and sterilization departments. Now, these are used maybe in between the times they use biological indicators, and we'll get into biological indicators next. But if they're not using or not required to use a biological indicator on every load, this would be used in between. But more popular for those in-between loads is definitely the type 5. And worldwide, type 5 seems to be the single most popular chemical indicator outside of the type 1 process indicators, because it is designed to react to all critical variables of time, temperature, and moisture, and their stated values. So the type 5 indicator is commonly used in sterilization validation as an internal indicator. So you put this in to an area where it might be within a load somewhere, or multiple loads. You may put one in every package you put into a load, to ensure that particular package or product was sterilized properly. And again, probably the most popular chemical indicator that we sell. The emulator, again, gaining popularity, but not quite as popular, certainly, as the type 5, but it's a little more stringent and will react to more critical processes for sterilization, and can be more precise for specific time and temperatures of specific sterilization programs. The Bowie-Dick test, again, it's considered a type 2, and it's again, it depends on what the requirement is of the facility or manufacturing facility company that runs these for this test. So it's used to verify the effectiveness of air removal in a pre-vacuum sterilizer. Again, if you haven't seen one of these before, it's a card that changes color. So you see the card there, and it's got pink. That'll change color, and that is inside the box behind it. That has like stacks of cardboard to challenge the device, so the validation is proper. So they put that card within like a deck of cardboard. Inside that box, you put the whole box in the sterilizer, and it will indicate whether or not the air removal was appropriate for in the pre-vacuum sterilizer. It indicates whether the sterilizer can be adequately remove air from the chamber, which is critical for effective sterilization. So biological indicators. Biological indicators are critical tools used to monitor and validate the effectiveness of sterilization processes that contain highly resistant microorganisms, usually bacterial spores, that are specifically selected to test the sterilization process ability to kill the most resilient microbes. And there's a host of different spores that are used for different processes. Okay. So, we can get into that, later, or if you look at some of our the literature that we have available. And I just wanted to point out now, all of our products, we have some digital brochures that outline in more detail what each product is, and those would be available to you upon request through the distributor. So key elements: spore forming bacteria, typically bacillus or geobacillus species, which are known for their resistance to sterilization methods. Care and packaging. BIs come in, and again, biological indicators come in various forms, such as spore strips, ampules, self-contained vials, which are placed inside or alongside the items being sterilized. And off to the side there, you can see we've got an ampule, we've got a self-contained biological indicator, and then the picture on the right are some steel discs that we would inoculate with spores. And then the growth medium, after exposure, the sterilization process, biological indicators are incubated in a growth medium to detect any surviving microorganisms. So that in some cases, like a self-contained biological indicator has the growth medium already in it, where others you would add it to the process upon incubation. So self-contained biological indicators, or SCBIs as they'll be referred to, we make them for steam and different populations of the spore, and that's why you have an 05 or an 06. We make a couple different types. We make a mini, which is our most popular, and that's the one on the top there. And then we make what we call a rigid, or it's a slightly larger version, and that's on the picture below. But I would say the majority of the ones we sell, probably over 75%, are what we consider our minis. Same thing for hydrogen peroxide, and then ethylene oxide has SCBIs as well. But for the most part, the majority we sell are the steam self-contained biological indicators. So I talked about how we will inoculate carriers with a particular spore, and this is what's probably the most prominent within, let's say, medical device industry, pharmaceutical industry in some cases. Anybody that's trying to either sterilize something in the process that they're doing for a project, or they want to see, or you want to see, if the device you manufacture, can it be sterilized, and can we validate that it can be sterilized? So we've got steel discs, but just like they sound, they're stainless steel. Spore strips, which is a paper strip that's inoculated. Spore threads, which essentially is about the size of dental floss, to be honest with you, and that can be put into small crevices. Paper discs, which we use, and we'll inoculate those. Spore wires, which are more rigid, it's more like a cable that will be inoculated, that can be put into different devices. And then spore ampules. So biological indicators are available on various carriers, each inoculated with the appropriate spore for specific sterilization method to be validated. A spore ampule is a type of biological indicator used to validate sterilization process, particularly steam and liquid. So you can drop that ampule in liquid. It consists of a hermetically sealed glass ampule containing a specific population of Geobacillus stereothermophilus spores suspended in tryptic soy broth. And that's just, I wanted to point that out because that kind of has everything already in it, all built into the ampule itself, where you're not going to be adding a medium later. Now, this is kind of a revolutionary thing. You may see in the market that some folks say out there they've got instant indicators, and in terms of their definition, I guess they do. But we actually are the only manufacturer in the world to have a true or instant indicator, and the instant indicator for steam. Right now, it is considered an enzymatic indicator as per the FDA. And we are working toward making this, giving it the category of a biological indicator. But it's so different than what the FDA was used to seeing that they have asked us to do additional testing to support that claim. But they said, yeah, this thing works, and it works fast. And just to give you an idea of how it works, you would put that into a load, and when you pull it out, you add a coenzyme to a tablet that's inside, and within 20 seconds, you get a validation result that yes, it was failed, or no, yes, it passed, or no, it didn't. Doesn't require any incubation. And the beauty of this is that this can be done like anywhere within a facility. You don't have to run and find an incubator or take it to a lab or send it somewhere. It can be done on site. And not to mention, once this becomes considered a biological indicator, at least for the U.S. market, this will speed up the getting the entrance back to, let's say, an OR, where they're standing up in the OR with a patient under anesthesia waiting for this to come back. This will give them an instant result once they get the sterilization process completed. And we're excited about this product, and we're more excited about getting our biological indication soon. But it's changed the way a lot of at least right at this point, we a lot with research labs and some in the waste disposal contamination. It's changed how they approach their business. So that's all I have. I hope I didn't talk too fast. I on my end, it seems like I'm talking really slowly, but I've been told I talk too quickly. So hopefully you got all that. I'm not sure if the process this presentation will be available to you. That's up to LASEC, but I'm here to answer any other questions. And I just wanted to let you know that I've got my chief science officer should be online as well. Just so you know, I am the sales director, where Tom Ria should be. I think he's on the call. He is the one that can answer any deep and technical product question that you might have. So
I am here.
Yes.
Oh, Tom, excellent. So I can answer. If did you want me to go through the first 3 questions that we received and go through those?
Yes. So we will now be moving over to the Q&A segment, where our attendees can ask any questions. The Q&A chat box is open for about 5 minutes, so please do try and submit your questions now. But we did receive about 3 questions before the webinar in our registration form. So those are the questions that Rick will be taking us through.
Okay, great. So first question we had was, how do chemical indicators complement biological indicators during sterilization monitoring, and what scenarios would you recommend using both together? That's a great question. And just to give you an idea of how they complement each other, all of our SCBIs have indicating ink on them. So we have a little indicating strip on all our self-contained biological indicators to say, yes, it was run through, or no, it wasn't run through the sterilizer. So that's just an indication of what, how important this can be, is to have both. But chemical indicators and biological indicators complement each other in the sterilization process by providing different types of information. Chemical indicators are used to monitor the presence and effectiveness of sterilization agent, like we talked about, steam, hydrogen peroxide, something like that. They change color when exposed to specific conditions such as temperature, time, and indicating that the sterilization agent has reached the necessary parameters. Again, doesn't say that it was effective, that it killed everything, but says yes, it ran through everything, and yes, the steam or the hydrogen peroxide did reach the area that we wanted to sterilize. So how do biological indicators fit into this? Well, they, like we talked before, they contain a highly resistant bacterial spore, and when placed in the sterilizer, well, the item's being sterilized, after the cycle, the BIs are incubated to see if any spores survive. If no growth is observed, it confirms the sterilization process was effective. So again, one will say, yes, the process, the gas, the steam got to the product, and yes, it was run through, but biological indicators are going to say, yeah, everything's dead. Everything's killed, or maybe it's not, and we've got to rerun the load or pull some instruments or something or devices from the shelves because it didn't work. But by using both together, you get a comprehensive assessment. CIs, or the chemical indicators, give you an immediate response. So you pull it out, yes, boom, we got it. Where biological indicators, we're going to take some more time to get that answer. Again, unless you use our instant, which I talked about. So, but note, some healthcare facilities require type 1 process indicators to be placed on all outer items being sterilized for immediate determination of processed and unprocessed goods, for the SPDs. Also, type 5 chemical indicators is to be exposed within every pack, tray, or cassette that is in a sterilization load, in addition to the BIs required for the cycle. So you may have some compliance, or some company compliance or healthcare compliance that's going to say you need to use both. Alternatively, some only require a BI once a week. Again, a biological indicator once a week to monitor sterilization, while the rest of the time you can use chemical indicators. So again, you might have a facility that says, hey, our standard is run it once a week with the biological. If it passes, the rest of the week you can run a chemical indicator. So that's the answer to that question. What are the best practices for positioning indicators in autoclaves to ensure accurate monitoring across different load types? Okay, positioning indicators correctly in autoclaves is crucial for ensuring accurate monitoring of the sterilization process. And here are some of the best practices. So a central placement of, placing the biological indicator and chemical indicators in the center of the load. This ensures that they are exposed to the most challenging conditions within the autoclave, providing a reliable measure of sterilization effectiveness. Multiple locations also can be used, and that's the use of multiple indicators and place them in different locations within the load. This helps to account for potential variations in temperature and steam distribution. And to further explain that, for instance, the US FDA approved biological indicators. They outline that they should position and or they position in the sterilizer will, hang on a second here. I apologize. I want to get it right from the FDA, because I'm always worried about reading FDA stuff. We'll outline where the BI should be positioned, and or the sterilizer manufacturer will outline this location. It's common during steam sterilization to have BIs near the drain, typically in the bottom or back of the chamber, as this is typically the worst location for them to be tested, due to location is usually at its lowest temperature during a given cycle. So that's kind of what the manufacturers, and that's what the FDA is saying is positioning of those. So again, it put it in the worst place, the most challenging place. And the last question, how many biological indicators should be used during sterilization? And is it necessary to use a mapping method with multiple indicators compared to a single point placement? The number of biological indicators used during sterilization can vary depending on the size and complexity of the load, as well as the specific sterilization process. However, a common practice is to use at least one biological indicator per sterilization load. For larger or more complex loads, it may be beneficial to use multiple biological indicators in different locations within the load to ensure comprehensive monitoring. Now, using a mapping method with multiple indicators is generally recommended over single point placement because it provides a more accurate assessment of the sterilization process. Multiple biological indicators can help identify any potential cold spots or areas where the sterilizing agent may not have penetrated effectively. This method ensures that the entire load has been exposed to the necessary conditions for sterilization. Now, the mapping method, I'm not sure who asked the question, but those who don't know, is where you identify the critical areas. So you determine the critical areas within the load where the sterilization might be most challenging, such as the center, corners, or bottom of the load. And then you place the multiple biological indicators in these critical areas. Using multiple biological indicators helps to account for any variations in temperature and steam distribution within the load. So that's what mapping is. So that's the answers to those first three questions.
Okay, great. We did also receive another question. Is chloride dioxide still used for decontamination, considering that it can sometimes be considered corrosive?
Tom, you want to take that?
Hey, can you repeat that again? What process?
Is chloride dioxide still used for decontamination, considering that it can sometimes be considered corrosive?
Yes, correct. Chlorine dioxide is still used today, but it is in very select markets. It is a very customized, specific sterilization method, most of the time in high-level disinfection, sometimes not always considered a sterilization cycle. We have a company here in the United States, chlorine dioxide is used, and there's another one out in Europe that utilizes it, and their cycles are very unique. And then some companies try to utilize them as well for the need of their product. But it is not a modality that has taken off, say, like hydrogen peroxide. But yes, it is corrosive, and it is very critical to know your material science of a given medical device when trying to use it.
Okay, and that brings us to the end of our webinar today. Thank you to the True Indicating team for their time and expertise, and thank you everyone for joining. To show how much we appreciate your attendance, we will be giving special discounts to all attendees. So please do reach out to your dedicated sales representative for more info. If you do have any further questions regarding True Indicating product and pricing, my email will be in the Q&A chat below, so please do feel free to reach out.